Welcome back to Cancer as a Mitochondrial Metabolic Disease Basics. Mitochondrial dysfunction doesn't happen all at once. It builds up over time. Mitochondria are constantly exposed to oxidative stress, and these small amounts of damage accumulate. Energy production gradually declines, and eventually, that shift can fundamentally change how a cell behaves. In this video, we'll explore how chronic mitochondrial insults can lead to mitochondrial DNA damage, how that damage accumulates over time, and how this progressive decline in mitochondrial function may contribute to the development of cancer. So without further ado, let's get into it. So this will be part 3, technically part 4 if we count the oncometabolites video, but part 3 in the what is mitochondrial dysfunction seen in cancer. And we left off on a very important slide. We talked about in the last video about the mitochondrial supercomplex and how it was important for energy efficiency, production, etc., and how when we lose efficiency, we end up with more oxidative stress, and that leads to further membrane damage and then further loss of the supercomplex, and how this becomes a very vicious snowball, and we lose ultimately energy production, which is critical for the cell to maintain itself. And we talked about how the mitochondrial cristae are critical to determine whether or not these supercomplexes can actually form and perform. And so I wanted to get into what is actually seen by researchers in the field in cancer when they're looking at mitochondria. So this is an electron micrograph picture of what a normal, quote unquote, mitochondria looks like, where you have the outer membrane, the inner membrane, and these cristae that are very linear and very organized appearing. And then this is a glioblastoma multiforme, for example, a brain cancer, where you have an outer membrane, you see an inner membrane. It looks diseased. And I am not an expert at electron micrographs, but I'll tell you that it doesn't look like a normal mitochondria does. I don't see any cristae, maybe cristae here. So therefore, there is either zero cristae or very, very disorganized cristae. And that brings me back to this slide here again, right? So if there is no cristae at all, we're not talking about just perturbed cristae. We're talking about no cristae at all. Do you think these respiratory complexes are going to be formed? And if there's no respiratory complexes formed, do you think we're going to be having any efficient oxidative phosphorylation? I'll leave that with you as an open question. Next graphic. Mitochondrial cristae of hypoxic cancer cells are wider, shorter, and less organized. And here again, you have normal mitochondria here. This is a different paper graphic. Normal mitochondria, you can see the cristae are there. And then here is just 72 hours of hypoxia. And you see here how the cristae are different shapes. You can see how the mitochondria themselves are different shapes, how they're different sizes, and how the cristae are much more disorganized after just 72 hours of hypoxia. And one thing we do know about cancer and tumors is that these cancer tumors are very hypoxic, on the order of 3 to 7 times more hypoxic compared to the normal tissues of origin. This is a graphic from a Seyfried paper looking at the structure of mitochondria in cancer. And again, you see diverse malfunctions, structural issues with these mitochondria. This is going to lead me to talk about a researcher at the Children's Hospital of Philadelphia named Dr. Doug Wallace. And he effectively created the idea of something called mitochondrial heteroplasmy. And what it effectively means is that there is varying ratios of healthy mitochondrial DNA inside mitochondria, and there are varying ratios of unhealthy DNA inside mitochondria. And that ratio of healthy to unhealthy mitochondrial DNA leads to OXPHOS, either efficiency or inefficiency. And when there's increasing mitochondrial DNA damage and mitochondrial structural damage, that leads to a bioenergetic decline that is progressive, and that leads to disease. And this channel is ultimately interested in having no disease whatsoever. I don't want there to be immunological diseases, neurodegenerative diseases, other metabolic diseases. But in this particular time, we're focusing on cancer. So cancer is also at the root of this progressive bioenergetic decline, as described by Wallace. So what does this look like on a time scale? So here is time 0. Let's say this is birth, and then this is time, let's say death. And ideally, again, this is a graphical representation of kind of ideal situations, but ideally, blue represents wild type or healthy mitochondrial DNA, and red represents mutant mitochondrial DNA or diseased mitochondrial DNA. And when we're born, ideally, we have very healthy mitochondrial DNA and mitochondria, because every mitochondria that's got more healthy DNA than less healthy DNA remains a blue, healthy mitochondria. And if the red or diseased mitochondrial DNA takes over as the dominant ratio of mitochondrial DNA, that becomes a red mitochondria in this graphic to illustrate what we're talking about here. Let's say, ideally, we all have 100% healthy mitochondria when we're born. That's unlikely to be the case, but let's just say, for simplicity's sake, we all are born with healthy mitochondrial DNA, which means that it's able to make healthy mitochondrial respiratory complexes, and ultimately you have efficient, healthy mitochondria. Energy production is normal or good, and the cell is happy, effectively. As we go through life, we accumulate mitochondrial DNA damage. And when that damage is lower, then the mitochondria will maintain OXPHOS efficiency, and it's going to overall contribute to an asymptomatic or disease-free situation. But as we go through time, we build up these mutations and more mitochondrial damage ensues, and ultimately we hit a biological threshold. And when we cross over this biological threshold, we now have an overall defective OXPHOS energy production through oxidative phosphorylation, and we developed a symptomatic problem, a symptom of some sort, a disease of some sort. What makes this process somewhat confusing is that although it's understood that you're going to develop about 10% heteroplasmy per decade, that is going to vary depending on the tissue, right? So if you have, I don't know, 50% or 60% in your kidney, you may only have 30% in your lungs, or maybe you have 70% in your pancreatic beta cells or your brain or whatnot. And then the location of that tissue dysfunction, because we have energy inefficiency, symptoms and disease are manifesting in that tissue, in that colony of mitochondria, that is where a disease will manifest. And that kind of explains why you would get, I don't know, progressive lung dysfunction by chronic insults from smoking, for example, or that smoking and the carcinogens that you take in hyperaccumulate in the bladder, let's say, and increases your risk for bladder cancer, specifically for smoking. So that kind of explains with a little bit more complexity and nuance, maybe how this really is happening. But this is kind of Wallace's framework for disease creation. And it's not just about mitochondrial DNA damage itself. It's also about potentially the amount and number of copies of DNA that could also be part of the disease process. So I pulled this paper from April of 2021, and it's titled Mitochondrial DNA Copy Number in Human Disease. The more, the better. And it says here in the abstract, most of the genetic information has been lost or transferred to the nucleus during the evolution of mitochondria. Nevertheless, mitochondria have retained their own genome that is essential for oxidative phosphorylation, OXPHOS. In mammals, a gene-dense circular mitochondrial DNA of about 16.5 kilobases encodes 13 proteins, which constitute only 1% of the mitochondrial proteome, or proteins made inside the mitochondria. Mammalian mitochondrial DNA is present in thousands of copies per cell, and mutations often affect only a fraction of them. Most pathogenic human mitochondrial DNA mutations are recessive and only cause OXPHOS defects if present over a certain critical threshold. That's the idea of mitochondrial heteroplasmy. However, emerging evidence strongly suggests that the proportion of mutated mitochondrial DNA copies is not the only determinant of disease, but also the absolute copy number that matters. In this review, we critically discuss current knowledge of the role of mitochondrial DNA copy number, regulation in various types of human diseases, including mitochondrial disorders, neurodegenerative disorders, and cancer, and during aging. We also provide an overview of new exciting therapeutic strategies to directly manipulate mitochondrial DNA to restore OXPHOS in mitochondrial diseases. So basically, it's not just about whether you have wild type mitochondria and the ratio of mutant mitochondria. It's also about how much overall mitochondrial DNA do you have in that cell. That's also a factor that's not taken into account in other publications and other schools of thought. So now I'm going to transition us to Professor Thomas Seyfried. And it's going to highlight a paper that he published with his group, of course, in November of 2020. And it's titled The Origin of ATP Synthesis in Cancer. And it says here, various anomalies in mitochondrial structure and function prevent maximal ATP synthesis through OXPHOS in cancer cells. Little ATP synthesis would occur through glycolysis in cancer cells that express the dimeric form of pyruvate kinase M2. Mitochondrial substrate-level phosphorylation, MSLP, in the glutamine-driven glutaminolysis pathway, substantiated by succinate-CoA ligase reaction in the TCA cycle, can partially compensate for reduced ATP synthesis through both OXPHOS and glycolysis. So what is Professor Seyfried saying? He's saying that various types of mitochondrial structure and functional problems limit the amount of ATP produced by cancer cells. Okay, that is completely in line with structure functional biology. That's completely in line with what Douglas Wallace is saying and the mitochondrial heteroplasmy people are saying. And we know that the structure of these cancer mitochondria are damaged. Okay, and we're going to look at how well that's been documented. So knowing that there's no cristae here and that there's little or no OXPHOS, of course, OXPHOS is going to be low in those mitochondria. What Seyfried is saying is that in order for these cells to survive, they have to upregulate a backup system of energy to survive. Otherwise, they're going to die, or at minimum, stop growing. So as energy is being lost by the progressive mitochondrial structural defects, which leads to functional defects, something has to take its place. And this is where the idea of what they call substrate-level phosphorylation is introduced as that backup system. You may have heard of this in a different way called the Warburg effect. Warburg effect is effectively cytosolic substrate-level phosphorylation. And in this paper, Seyfried is referring to another form of substrate-level phosphorylation called mitochondrial substrate-level phosphorylation. And that is where, despite having structural differences in the cristae and OXPHOS system, the TCA cycle is still operational, does not require oxygen. And this is where glutamine can be fermented into energy as well to make up for the loss of OXPHOS and any limitations that could be of the glycolysis or the burning of glucose as ATP. And so we see here something that is very similar, strikingly similar to what Wallace and others are saying, is that we have this progressive bioenergetic decline, right? And what we're seeing from Seyfried is adding a layer of complexity is that something has to make up for that energy loss. And OXPHOS is very efficient in making energy. One molecule of glucose will make 36 ATP, whereas one molecule of glucose for this SLP system will only make 2. So it needs to bring in a lot more glucose and glutamine to make up for that loss. And that's what this graphic is showing. As OXPHOS efficiency goes down, those other backup systems are upregulated in the cells that survive. And that's how the energy flux transition happens. It's very likely not immediate. It's very likely happening over time. And so what Seyfried has put in these graphics are basically words without words, right? So you see here, this mitochondria looks structurally intact. You see the mitochondrial cristae. They look very formed. What do you see as time goes on? You see the cristae being less formed and even non-existent, right? So this is what is happening over time. This is the graphical representation of this. This is before and after, right? But this is what's happening over time. Because as we lose these cristae, as we know, can no longer have efficient electron transfer, we have decreased OXPHOS, and we have more oxidative stress and lipid peroxidation in that vicious snowball. This is the vicious snowball in action. What does he have here also? He has genes and inflammation and oxidative stress and various ways that mitochondria can be damaged over time. This is what kind of how carcinogens effectively damage the cell. It's mostly thought of in biology that these carcinogens are damaging these nuclear DNA. And I don't think Seyfried is saying that there are not known somatic DNA mutations in the nucleus that are damaged. They can be, but they also, in some cancers, are not there, which makes it probably less of an important piece of this whole thing than this, okay? Because these are there axiomatically there. So my point is that these carcinogens and these damages that accumulate from a variety of sources over our lifetime damage these mitochondria, lead to a bioenergetic decline, and in certain cells that have the ability to, at the same time, stabilize their energy requirements through these backup systems, survive, and then become cancer. This is another paper that was more recently published in July of 2023 and is titled Role of Mitochondrial Alterations in Human Cancer Progression and Cancer Immunity. And it says here, dysregulating cellular metabolism is one of the emerging cancer hallmarks. Mitochondria are essential organelles responsible for numerous physiologic processes, such as energy production, cellular metabolism, apoptosis, and calcium and redox homeostasis. Although the Warburg effect, in which cancer cells prefer aerobic glycolysis, even under normal oxygen circumstances, was proposed a century ago. How mitochondrial dysfunction contributes to cancer progression is still unclear. This review discusses recent progress in the alterations of mitochondrial DNA and mitochondrial dynamics in cancer malignant progression. Moreover, we integrate the possible regulatory mechanisms of mitochondrial dysfunction-mediated mitochondrial retrograde signaling pathways, including mitochondrion-derived molecules, reactive oxygen species, calcium oncometabolites, and mitochondrial DNA. The mitochondrial stress response pathways, mitochondrial unfolded protein response, and integrated stress response in cancer progression provide the possible therapeutic targets. Furthermore, we discuss recent findings of the role of mitochondria in immune regulation function of immune cells and reveal the impact of the tumor microenvironment and metabolism remodeling on cancer immunity. That's kind of a lot to unpack. So let's take a look at this graphic that is associated with this paper. We see here that there are mitochondrial alterations, and those alterations can be in the way that mitochondrial life cycle is happening. That's what they're talking about with mitochondrial dynamics, fission, fusion, mitophagy, mitochondrial biogenesis, etc. There's life cycles of the mitochondria. And there are mitochondrial DNA alterations. There's our heteroplasmy. Remember, also mitochondrial DNA amounts has a role here too. Ultimately, these things can lead to an overarching mitochondrial dysfunction, bioenergetic decline. Then we lose control of calcium regulation, oncometabolites, reactive oxygen species, which can damage nuclear DNA also through excessive mitochondrial retrograde signaling. And that can lead to further progression and formation of disease. In this case, we're talking about cancer. So let's look at this from a higher level or from a different perspective. So this is a non-tumor oxidative cell, which means there is oxygen present and these mitochondria are intact. So they are metabolically flexible and they are able to make energy through oxidative phosphorylation. However, as we accumulate damage, the ability for the mitochondria to use oxygen effectively and macronutrients effectively to make energy is deficient and or ineffective. So what takes over is an upregulation of glucose transporters and the machinery that's needed to ferment glucose and glutamine without oxygen that don't require intact mitochondrial structure. And this will take over what we're losing here. So this is kind of the midpoint here. This is kind of like here in this area, right in the middle. Now, at the end of this, over here, or in a Seyfried graphic over here on this side of it, where we have completely dysfunctional mitochondria and we have hypoxia, a low amount of oxygen, we have severe mitochondrial defects, which means that there is low or no OXPHOS being made, which should answer the question already, can these cancer cells burn fat for fuel? Can these cancer cells burn ketones for fuel, et cetera, et cetera? Handle that for another day. But the bottom line is these mitochondrial defects are so severe that they are unable to make energy. So what the cancer cells actually do is they'll actually shut down normal energy flux through mitochondria, and they will divert all of the flux of energy production through aerobic glycolysis, or the Warburg effect, or as what Seyfried is now calling cytosolic substrate-level phosphorylation, and glutaminolysis, or the breakdown of glutamine, the amino acid glutamine, and making energy without oxygen in that pathway too, and that's called mitochondrial substrate-level phosphorylation. So that's what Dr. Seyfried in this paper we talked about, substrate-level phosphorylation, is talking about. As OXPHOS efficiency goes down, the utilization of these other backup systems of energy production, what he calls substrate-level phosphorylation, is now trying to specifically call mitochondrial and cytosolic substrate-level phosphorylation are taking over as the predominant form of energy produced by these cancer cells as a way to survive. They're just trying to survive. So you may be asking yourself, am I, and is my type of cancer that I'm dealing with, or that my family member or my loved one is dealing with, is it under this theory? Is it operational under this theory? Well, Dr. Seyfried, in a very important paper in his group, published this table here. And this table is basically, probably without making the table too extensive, nearly every observable human cancer. And what he did, and his group did, is they documented every paper with little hyperlinks here, so you can click on it and find that paper, of where bladder cancer has been shown to have mitochondrial abnormalities seen. Okay, or cancer has mitochondrial abnormalities seen, or colorectal cancers, or gliomas, or non-solid tumors like AML, CLL, ALL, renal cell carcinoma, hepatic cancer, lung cancer, melanoma, and down and so on and so forth. So this is operational in every observed cancer that Seyfried and others have studied. So therefore, the mitochondrial metabolic theory of cancer, the progressive bioenergetic loss and of the use of OXPHOS is operational in every cancer. And that's due to observed mitochondrial structural defects, which leads to functional defects, which means that OXPHOS cannot happen, even if oxygen was present, which is what the Warburg effect means. We also know that this is compounded by the fact that the tumors also are hypoxic. They do not have oxygen either. So even if Wallace and others, even if Seyfried and his group, and all these other researchers that have found that there are dysfunctional mitochondria, structural dysfunction in all of these cancers were wrong. Let's say, I don't know, 100 people now, and all their groups are wrong. Even if they were wrong, these tumors are known to be hypoxic, and that by itself would lead to this problem. So the reality is we know that both are true, that we have mitochondrial observed structural defects, which leads to functional defects, and we have hypoxia. So those two facts is what has led those like Seyfried to propose something called metabolic therapy for cancer as a treatment modality based off these facts and based on these are known hallmarks of all cancers, and they are known metabolic differences between normal cells, right? Normal cells have good mitochondria, they have oxygen, they are metabolically flexible, and these cancer cells are not metabolically flexible, they don't have good mitochondria, and they don't have oxygen. So when we know what separates them from us, then we can use those differences against them. And that is the basis for not only the mitochondrial metabolic theory of cancer, but it's also the basis for ketogenic metabolic therapy or press-pulse metabolic therapy as also proposed by Seyfried. If you like videos like this, please like, share, and subscribe. And until next time.